alimentary canal. The 9-meter-long alimentary canal is a tubular structure composed of the esophagus, stomach, small and large intestines, and anal canal. It digests food, absorbs nutrients and water, and compacts and eliminates the indigestible components of the ingested food. General organization of the alimentary canal. The digestive tract is composed of concentric cylinders around a lumen. These layers are modified along the canal, but before discussing the modifications, the general pattern is described. Fig 17.1. The lumen is lined by an epithelial layer and a subepithelial connective tissue, the lamina propria, which houses glands and lymphatic nodules that constitute the mucosa-associated lymphoid tissue. The lamina propria is surrounded by the Muscularis mucosae, two smooth muscle layers arranged in a helical fashion, an inner circular and an outer longitudinal layer. The epithelium, lamina propria, and Muscularis mucosae together are the mucosa. Surrounding the mucosa is a dense, collagenous connective tissue, the submucosa, which is a vascularized region that houses glands but only in the esophagus and duodenum. Meissner submucosal plexus, a component of the enteric nervous system, occupies the most peripheral layer of the submucosa, serving intramural glands, vascular supply and Muscularis mucosae, and acts locally. The Muscularis Externa is composed of smooth muscle in two helically disposed layers, an inner circular and an outer longitudinal layer. Auerbach's, my enteric, plexus lies between these two smooth muscle layers and controls peristalsis. Auerbach's plexus acts locally and also over long stretches of the alimentary canal. The alimentary canal is covered by either connective tissue, adventitia, which affixes regions of the gut to the body wall, or by a moist simple squamous epithelium, serosa, which reduces friction as the gut moves during peristalsis. The enteric nervous stem can act completely on its own, however, it is modulated by the sympathetic and parasympathetic nervous systems. The esophagus, a 25 cm long muscular tube whose wall is collapsed unless it is transmitting a bolus into the stomach, closely follows this general plan. The esophageal mucosa is composed of a stratified squamous non-keratinized epithelium. A lamina propria whose esophageal cardiac glands produce mucus that aids in swallowing the bolus. These glands are located in the regions near the pharynx and near the stomach. The Muscularis mucosa is composed of a longitudinally disposed smooth muscle layer. The vascular submucosa has the esophageal glands proper, which produce mucus and serous secretions. The serous component of this gland manufactures pepsinogen, a proenzyme, and lysozyme, an antibacterial agent. The mucus component lubricates the epithelium. The esophagus has glands in its submucosa. The Muscularis externa, composed of an inner circular and an outer longitudinal layer, is unusual because in the upper one-third of the esophagus, near the pharynx, both layers are composed of skeletal muscle. In the middle third, they are composed of skeletal and smooth muscles, and in the lower third, near the stomach, they are composed solely of smooth muscle. The outermost layer of the esophagus is the adventitia in the thorax and a serosa once it enters the abdominal cavity. The most common symptoms that are indicative of disorders of the digestive system include dysphagia, problems with swallowing, regurgitation, constipation, diarrhea, and bleeding from the digestive tract. Dysphagia may have numerous causes, including physical obstructions in the pharynx or esophagus, muscular or neural problems, and psychogenic problems. Regurgitation is not accompanied by nausea or by violent constriction of the abdominal musculature that occurs during vomiting. The process of regurgitation may be due to neural or muscular problems associated with the esophageal sphincter or due to stenosis of the esophagus as a result of a malignancy or untreated acid reflux. Regurgitation in the absence of a physical cause is known as rumination. In this condition, the swallowed food returns to the mouth, where it may be chewed and swallowed again. Rumination is common in infants, it may occur in adults also and is usually stress-related. Constipation is a condition in which bowel movements occur less than 3 times per week. It is more common in women than in men, in adults older than 65 years, and in women who are pregnant. Individuals who are constipated usually produce small, dark, dry, Hard stools that are difficult to eliminate. The causes of constipation may be dietary, example low fiber consumption, eating too much dairy, not drinking enough fluids, sedentary lifestyle, certain medications, including overuse of laxatives, disruption of the daily routine as in traveling, and ill health, such as stroke and intestinal disorders. Diarrhea refers to the production of loose, watery stool at least 3 times in one day. There are 2 types of diarrhea: acute and chronic. Acute diarrhea is quite common, lasts for 1 or 2 days, and resolves itself. If it lasts for more than 2 days, it is considered to be chronic diarrhea, and a physician should be consulted to prevent dehydration and rule out organic disease. Most cases of acute diarrhea are due to bacterial, viral, or parasitic infections, whereas chronic diarrhea is usually due to problems with the alimentary canal, such as irritable bowel syndrome. Bleeding from the digestive tract may occur from the mouth as bloody vomiting or from the anus as bloody discharge or bloody stool. The blood may be fresh or coagulated. Usually, if the blood is fresh, it is red in color and originates near the oral cavity or the anus. If the blood is coagulated, it appears as black particulate matter that resembles coffee grounds in the vomit, or it stains the stool black, melena. Bleeding from the alimentary canal may be caused by peptic ulcers, varices that leak blood, use of certain anti-inflammatory agents, inflammatory bowel disease, and cancer. The stomach has an inlet, where the esophagus joins it, and an outflow, where it is joined by the duod number. It can expand from a 50 ml volume when empty to approximately 1,500 ml when distended. As the stomach receives a bolus from the esophagus, it secretes gastric juices to liquefy the bolus into an acidic fluid known as chyme and to begin to digest it via hydrochloric acid and its enzymes, renin, pepsin, and gastric lipase. The hormone ghrelin maintains a constant intraluminal pressure by allowing the muscularis externa to adapt to the expanding volume and sustains the feeling of hunger as the stomach is being distended. The acidic chyme is released in 1 to 2 ml aliquots into the duodenum through the pyloric sphincter, the modified inner circular layer of the muscularis externa. The stomach has a pyloric region at the greater curvature, a cardiac region at the concave lesser curvature, 2 additional anatomic regions, the fundus and the body, which are identical histologically and are referred to as the fundic region. The lumen of the empty stomach presents rugae, folds of the mucosa and submucosa, which disappear when the stomach is distended. The stomach lining displays numerous epithelially lined depressions, gastric pits, foveolae, whose floor is perforated by many tubular gastric glands populating the highly vascular lamina propria. The Muscularis mucosae has 3 layers of smooth muscle: inner circular, outer longitudinal, and a poorly defined outermost oblique layer. The submucosa is unremarkable. The Muscularis externa has inner circular, outer longitudinal, and innermost oblique layers. The simple columnar epithelium of the fundic stomach is composed of tightly packed surface lining cells and regenerative cells. The lateral cell membranes of these cells form tight junctions with each other. Surface lining cells produce the thick, visible mucus that guards the epithelium from the acidic chyme, and regenerative cells proliferate to replace the stomach's epithelial lining. The gastric pits of the fundic and cardiac regions extend 1/3 of the way into the lamina propria, which is crowded with gastric glands. Each gland possesses 6 cell types, distributed disproportionately, in its 3 regions: the isthmus, which pierces the gastric pit, the neck, and the base, which abuts the muscularis mucosae. The gastric pits of the pyloric region extend halfway into the lamina propria. Cellular composition of fundic glands: mucous neck cells manufacture soluble mucus that becomes part of the chyme and lubricates the alimentary canal. The plasmalemmae of these cells form tight junctions with their neighbors. The regenerative cell's rapid mitotic rate replaces the entire epithelial lining every 5 to 7 days. Parietal, oxyntic, cells are usually not present in the base of the gland. They have deep intracellular canaliculi that are lined by microvilli. An intracellular vesicular network, the tubulovesicular system, whose membrane component is rich in the proton pump Hk, At posse, parallels the intracellular canaliculi. Parietal cells produce hydrochloric acid and gastric intrinsic factor. During Hcl production, the tubulovesicular system is reduced in size with a concomitant increase in the number of microvilli, suggesting that the vesicular network is used to store membranes destined for microvillus production, enabling the cell to increase its surface area during its secretory activity. With the cessation of Hci production, the microvillus membranes are returned to the tubulovesicular system. This energy-requiring process is fueled by the abundant mitochondrial content of parietal cells. The glycoprotein gastric intrinsic factor is released into the lumen of the stomach, where it complexes with vitamin B and to be absorbed by cells of the ileum. Chief, zymogenic, cells, located mostly in the base of fundic glands and not at all in cardiac or pyloric glands, manufacture pepsinogen, renin, and gastric lipase. The proenzyme pepsinogen is converted to the proteolytic enzyme pepsin in the acidic milieu of the stomach. Renin, chymosin, is a proteolytic enzyme that curdles milk, and the enzyme gastric lipase degrades lipids. These enzymes are present within vesicles in the apical cytoplasm of chief cells and are released because of the interaction of acetylcholine and secretin as they bind to their respective receptors on the basal plasma membrane of chief cells to activate their second messenger systems. Diffuse neuroendocrine system, DNES, cells are of two types: open and closed, where the former reach the lumen, and the latter do not. Each DNES cell produces a particular hormone that it releases into the lamina propria. These hormones are autocrine if destined for the releasing cell, paracrine if destined for a cell nearby, and endocrine if they have to travel via the bloodstream. Gastric histophysiology: on a daily basis, the glands of the stomach produce about 2 to 3 L of secretions whose main constituents are water, HCl, enzymes, intrinsic factor, and soluble and visible mucus. By coating the lining of the stomach, the neutral pH of the visible mucus protects the stomach lining from the acidic gastric juices. The visible mucus also affords a beneficial environment for H. pylori, the bacterium that resides within it. The Muscularis Externa of the stomach functions in churning and liquefying ingested food, forming a thick chyme that resembles split pea soup. The Muscularis Mucosa ensure that the entire epithelial surface of the stomach comes into contact with the chyme. When the chyme is of proper consistency, and depending on its acidity, osmolality, and lipid and caloric content, DNES cells of the duodenum release the hormone gastrin, which induces the pyloric sphincter to open and the longitudinal muscles of the pylorus to contract, injecting 1 to 2 ml of chyme into the duodenum. If the chyme is not ready to be discharged, DNES cells release the hormones cholecystokinin and gastric inhibitory peptide, which inhibit the pyloric sphincter from opening, and the chyme remains in the stomach. Hcl production in the stomach has 3 phases: 1 cephalic, referring to the thought, smell, or sight of food. 2 gastric, the presence of food in the lumen of the stomach. 3 and intestinal, the presence of food in the duodenum. The mechanism of Hcl production by parietal cells is the same, however, regardless of the phase that induced it. The basal aspect of the cell membrane of parietal cells possesses receptors for the neurotransmitter acetylcholine and for the paracrine hormones gastrin and histamines. When all 3 sites have bound their respective signaling molecules, Hcl production and its secretion into the intracellular canaliculi occurs in the following manner: carbonic anhydrase, present in the cytosol, catalyzes the production of HCO, which dissociates into H and Hc3 degrees active transport exchanges intracellular. H** for extracellular K located in the intracellular canaliculi. Although K is also actively transported into the parietal cell at its basal cell membrane, it also leaves the parietal cell via K channels located in the basal plasmalemma. K and Cl are actively transported out of the cell and into the intracellular canaliculi, where H** and Cl combine to form HCl. The circulation of ions between the parietal cell and the extracellular fluid of the lamina propria alters osmotic pressures, causing the flow of Ho into the parietal cells. The movement of ions between the parietal cell and the intracellular canaliculi alters the osmotic pressure within the parietal cell, causing the movement of HO into the intracellular canaliculi. HC1 and water, the main components of the gastric juice, are released into the intracellular canaliculi, spaces that are continuous with the lumen of the stomach. The presence of tight junctions formed by the epithelial lining of the gastric mucosa prevents the entry of HCl from the gastric lumen into the lamina propria. Additionally, the HCO* that is produced by the parietal cell is released into the lamina propria and, by its buffering action, protects the lamina propria from the accidental leakage of HCl from the gastric lumen. Another possible protection is afforded by the release of prostaglandins in areas whose epithelial barrier is accidentally breached. Prostaglandins in the lamina propria amplify blood flow to the area, facilitating the elimination of H from the affected region. The inhibition of Hcl production and secretion is controlled by 4 hormones: prostaglandin, gastric inhibitory peptide, and urogastrone act directly on parietal cells, inhibiting their secretory activity. The 4th hormone, somatostatin, inhibits the release of gastrin by C cells and histamine by enterochromaffin-like cells, eliminating the signaling molecules necessary for inducing parietal cells to make and secrete HCl. Small intestine. The small intestine is approximately 7 m in length and is said to have 3 regions. The first portion is the very short duodenum, about 25 centimeters long. The middle portion is the jejunum, whose wall is relatively thick and is a little less than 3 m long, and the third portion is the ileum, which is the narrowest, has the thinnest walls, and is about 4 m in length. The small intestine receives digestive enzymes from the pancreas and bile from the gallbladder, which assist it in digesting the food in its lumen and absorbing water and the nutrients generated. Histologically, the 3 regions are quite similar to each other. After a general description of their common features, the differences among the 3 regions are described in detail. Common histologic features: to increase the luminal surface area of the small intestine, its submucosa and mucosa have transverse folds, plicicirculars, valves of Kirkring, begin in the duodenum and extend into the ileum, retard the velocity of chyme, and augment the surface area 2 to 3-fold. Finger-like extensions of the lamina propria, villi, are covered by a simple cuboidal epithelium and enlarge the luminal surface 10-fold. These villi are 1.5 mm tall in the duodenum, 1 mm tall in the jejunum, and 0.5 mm tall in the ileum. Each villus possesses a vascular, loose connective tissue core with a blindly ending lymph capillary, the lacteal. Simple columnar epithelial cells covering each villus possess abundant microvilli that augment the luminal surface area 20-fold. Intervillous spaces of the small intestine display openings of the crypts of Lieberkühn, which increase the luminal surface area by a factor of 3 to 4. Histology 0F. The small intestine. The mucosa of the small intestine comprises the epithelial lining, lamina propria, and Muscularis mucosae, Fig 17.5. The simple columnar epithelium covering the villus is composed of surface absorptive cells. The most abundant of the cells function in the end stage of digestion and in absorbing amino acids, lipids, and sugars. These cells possess 3,000 microvilli covered with a glycocalyx. The glycocalyx comprises mostly enterokinases, aminopeptidases, and oligosaccharidases, enzymes that digest oligopeptides and oligosaccharides. The lateral aspects of the cell membranes of surface absorptive cells adhere to the membranes of adjacent cells by forming junctional complexes. Goblet cells manufacture mucinogen, a complex protein polysaccharide that, when in contact with water, becomes mucin. When released into the lumen, it mixes with the luminal content and becomes a slippery substance known as mucus. DNES cells constitute about 1% of the villus epithelial cells, each producing a specific paracrine/endocrine hormone. Microfold cells, M cells, located where lymphoid nodules of the lamina propria contact the epithelium, have deep folds intercellular pockets. M cells phagocytose intraluminal antigens and transfer them to lymphocytes present in their intercellular pockets, which then transfer the antigens to APCs of the lamina propria to initiate an immune response. Some of the IgA manufactured by plasma cells is endocytosed by epithelial cells that couple secretory component to it and release the complex into the lumen. Most of the IgA travels to the liver, hepatocytes complex it with secretory component and release it into the bile to be transported to the gallbladder. The lamina propria of the mucosa is a loose connective tissue that is rich in lymphoid and capillary elements, and, in the core of the villus, possesses lacteals. The deeper aspect of the lamina propria, between the base of the villi and the Muscularis mucosae, is quite vascular, although it is mostly displaced by the abundance of intestinal intramural glands, the crypts of Lieberkühn. These glands extend from the intervillous spaces to the Muscularis mucosae, and their epithelium consists of the same cell types as those covering the villus, and regenerative cells proliferate to form new cells of the epithelial lining. The new cell migrates along the basal lamina to the tip of the villus, where it is sloughed off into the lumen 5 to 7 days after its formation. The epithelial lining of the small intestine is replaced once every week. Paneth cells live longer, 20 days, are located at the base of the crypts of Lieberkühn, and house large eosinophilic granules that contain lysozyme and defensins, antimicrobial agents, and tumor necrosis factor Ti. The Muscularis mucosa is composed of an inner circular and an outer longitudinal layer of smooth muscle. Occasional smooth muscle cells enter the core of the villus extending to its tip. The submucosa of the small intestine is unremarkable, and its Muscularis externa is composed of an inner circular and an outer longitudinal layer. Meissner's and Auerbach's plexuses occupy their normal positions. The outermost layer is a serosa except in parts of the duodenum where it is an adventitia. Defensins are produced by Paneth cells, in response to tumor necrosis factor A and because of the presence of metabolic byproducts released by microorganisms. Defensins insert into the phospholipid membranes of microorganisms, where they form ion channels that permit the leakage of ions from the invading organism, killing it. Lysozyme, a proteolytic enzyme also manufactured by Paneth cells, breaks down the bacterial membrane component peptidoglycan, killing the organism. Regional differences in the histology of the small intestine. The three regions of the small intestine may be differentiated from one another by minor variations in their histologic appearance. The villi are tallest in the duodenum, 1.5 mm, shorter in the jejunum, 1 mm, and shortest in the ileum, 0.5 mm. The major difference is the presence of Brunner's glands in the submucosa of the duodenum. Peyer's patches, which are collections of lymphoid nodules, are present in the lamina propria of the ileum. The jejunum has neither glands in its submucosa nor Peyer's patches in its lamina propria. The duodenum receives the bile duct and pancreatic duct at the duodenal papilla, papilla of Vater. Brunner's glands, duodenal glands, are tubulo-alveolar, branched glands that open into the base of the crypts of Lieberkühn. Brunner's glands manufacture a seromucous fluid rich in bicarbonates that acts to buffer the acidic chyme delivered from the stomach. These glands also manufacture the hormone eurogastrone, human epidermal growth factor, which inhibits parietal cells from manufacturing HCl and stimulates the mitotic activities of epithelial cells. Histophysiology 0F. The small intestine. The motility of the small intestine has two phases: mixing contractions, which are localized events that function in exposing the luminal contents to the epithelial lining of the gut, and propulsive contractions, peristaltic waves, which move the luminal contents along the length of the small intestine. This movement is slow, about 1 to 2 centimeters per minute, and chyme entering the duodenum from the stomach spends 6 to 12 hours in the small intestine. The propulsive contractions are controlled by Auerbach's plexus and by the DNES-derived hormones cholecystokinin, gastrin, motilin, substance P, and serotonin, which increase motility, and secretin and glucagon, which retard motility. Glands of the small intestine secrete 2L of a seromucous secretion on a daily basis. The secretory process is mostly under the control of Meissner's submucosal plexus, but also is influenced by the DNES-derived hormones secretin and cholecystokinin. Digestion of the food material present in the lumen of the small intestine is due to the presence of enzymes, mostly derived from the pancreas, and bile from the liver, and concentrated in and delivered from the gallbladder. The process of digestion begins in the oral cavity and stomach and continues in the duodenum, where pancreatic enzymes degrade the various components of the chyme into oligopeptides and dipeptides and oligosaccharides and disaccharides. Enterokinases and aminopeptidases, located in the glycocalyx of the microvilli, complete protein digestion into amino acids and dipeptides and tripeptides that are absorbed into the surface absorptive cells to be converted into amino acids by cytoplasmic peptidases. The glycocalyx also contains oligosaccharidases, lactase, maltase, sucrase, and acetyldextrinase, that complete the digestion of dietary carbohydrates by degrading oligosaccharides into monosaccharides that are absorbed by the surface absorptive cells. Lipids present in the chyme are emulsified by the bile salts into micelles, and the pancreatic lipase breaks down the lipids into fatty acids and monoglycerides, which diffuse through the plasmalemma of the microvilli. Absorption of the end products of digestion and electrolytes and water occurs mostly in the small and large intestines, although certain substances, such as alcohol, are also absorbed in the stomach. Enormous quantities enter the surface absorptive cells of the small intestine, including 1 kg of fat, 0.5 kg of proteins and carbohydrates, about 35 g of sodium, and 7 L of fluid per day. Amino acids and monoglycerides are released into the core of the villus, enter the tributaries of the hepatic portal vein, and go to the liver for further action. Monoglycerides and long-chain fatty acids bind to intracellular fatty acid-binding proteins, and enter the smooth endoplasmic reticulum to be esterified by acyl-CoA synthetase and acyltransferases into triglycerides. These triglycerides, coupled to proteins, are transported to the basolateral membrane to be released into the core of the villus as chylomicrons that enter the lacteals, which become filled with the lipid-rich fluid, chyli. The lacteal is emptied because of the rhythmic contractions of the slips of smooth muscle derived from the inner circular layer of the Muscularis Mucosae, discharging the chyli into the submucosal lymphatic plexus. The chyli is transported to the thoracic duct to be delivered into the junction of the subclavian vein with the left internal jugular vein. Fatty acids that are less than 12 carbons long avoid the reesterification process. Instead, they pass to the basolateral cell membrane to be released into the core of the villus to travel to the hepatic portal vein and from there to the liver. Large intestine. The large intestine, approximately 1.5 m long, comprises the cecum, appendix, colon, rectum, and anus. Histologically, except for the appendix and the anus, these regions are very similar and are referred to as the colon. The colon functions in the absorption of water, electrolytes, and gases, and in the compaction of chyme it receives from the ileum into feces. The colon resembles the small intestine except that it has a greater diameter and possesses no villi. The crypts of Lieberkuhn resemble their counterparts in the small intestine, but they lack Paneth cells, possess few DNES cells, and have a greater number of goblet cells. Although it would appear at first glance that most of the epithelial cells are goblet cells, actually more than half of them are surface absorptive cells. As in other regions of the alimentary canal, the entire epithelial lining is replaced via mitotic activity of the regenerative cells at least once per week. The Lamina Propria Muscularis Mucosa and submucosa are unremarkable. The Muscularis Externa is modified in that many of the smooth muscle fibers of the outer longitudinal layer are collected into 3 slim bands of muscle, the tenui coli, that are almost in constant tonus, making them shorter than the colon. The colon forms a linear sequence of pouches, called haustra coli, along its length. The entire colon is invested by a serosa except at the anus, where it is attached to the body wall by a connective tissue adventitia. Along the length of the colon, the serosa forms fat-filled pouches known as appendices epiploicae. The function of the colon is the secretion of bicarbonate-rich mucus. It also absorbs more fluid and electrolytes from the intestinal contents, accomplishing the compaction of the feces. Every day, the colon reclaims about 1.4 L of electrolyte-containing fluids and reduces the daily feces volume to approximately 100 ml. The colon absorbs approximately G to 9 L of gases a day, releasing only about 0.5 to 1 L of gas as flatus. The crypts of Lieberkühn of the rectum are sparsely distributed and deeper than those of the colon, otherwise, the rectum greatly resembles the colon. Anal canal. The 3 to 4 cm long anal canal is narrower than the rectum, and in its lower half it does not possess even the shallow crypts of Lieberkühn present in its upper half. The anal mucosa presents longitudinal folds, anal columns, of Morgagni, that converge at the pectinate line to form the anal valves that house the pocket-like anal sinuses. The anal canal is lined by a simple cuboidal epithelium that becomes stratified squamous non-keratinized below the pectinate line. The fibroelastic lamina propria houses circumanal glands at the anus, hair follicles and their attendant sebaceous glands are present here. The Muscularis mucosae is present but does not extend past the pectinate line. The fibroelastic connective tissue of the submucosa of the anal canal has an internal hemorrhoidal venous plexus above and external hemorrhoidal venous plexus below the pectinate line, just above the anal orifice. The Muscularis externa is unremarkable except that the inner circular layer is thickened at the pectinate line to form the internal anal sphincter muscle, and the external longitudinal layer is replaced by a fibroelastic membrane that surrounds the internal anal sphincter. The external anal sphincter muscle is formed by thickenings of the skeletal muscle of the floor of the pelvis and surrounds the internal anal sphincter and the fibroelastic sheath. This skeletal sphincter muscle permits voluntary control over the anus. The outermost layer of the colon is a serosa. The appendix. The appendix is a narrow 5 to 6 cm long outpocketing of the cecum whose stellate lumen is lined by simple columnar epithelium composed of surface absorptive cells, goblet cells, and M cells that adjoins the lymphoid nodules present in the lamina propria. The crypts of Lieberkühn are sparse and shallow and are composed of surface absorptive cells, goblet cells, regenerative cells, numerous DNES cells, and occasional Paneth cells. The lamina propria is a loose connective tissue richly endowed with lymphoid cells and lymphoid nodules. The Muscularis mucosae, submucosa, and Muscularis externa follow the common organization of the digestive tract. The outermost layer of the appendix is a serosa. Crohn's disease. Crohn's disease is a chronic inflammation of the wall of the alimentary canal that occurs most frequently in the ileum and the colon. It is believed to be an immune-related disorder that may have genetic and environmental components. It occurs in equal distribution among men and women, making its first appearance usually between ages 15 and 25 years, but almost always before age 35. The usual symptoms are fever, chronic diarrhea that may or may not be accompanied by bleeding, abdominal cramps of varied severity, weight loss, and lack of appetite. The symptoms may last several weeks and then resolve by themselves, only to recur at various indeterminate intervals with variable degrees of severity. The location of the inflammation may be the same as before, or it may spread to other regions of the alimentary canal. Common complications are the formation of fistulas and obstruction of the digestive canal, and in the colon it may lead to colorectal cancer. Although Crohn's disease is incurable, palliative treatments include the use of antidiarrheal and anti-inflammatory agents, antibiotics, immunomodulators, dietary modifications, and, if necessary, surgical resection of the affected areas. Cholera. Individuals with cholera have ingested water or food infected with Vibrio cholerae, an organism that produces cholera toxin. Although cholera is a very easily treated disease, it is widespread in the tropical and subtropical regions in developing countries, where it is responsible for a high degree of fatality. Cholera is prevented successfully by employing proper sanitary conditions and is treated by antibiotics and the rapid administration of electrolyte-balanced fluids to replace fluid and electrolyte losses, which may be 10L/day owing to uncontrolled diarrhea. Intestinal gas. Intestinal gas is a byproduct of bacterial metabolism and some swallowed air. The odoriferous components of feces are mercaptans, indole, and hydrogen sulfide, and the odoriferous component of flatus is methane. Because the methane gas is mixed with Co and He, it is quite flammable, and occasionally during cauterization as part of sigmoidoscopy, small, localized explosions may occur.